Amino acids and proteins. We need a variety of foods every day to keep us going. Foods rich in proteins help us to grow and heal. But how do we know which foods contain protein? We're going to answer this question in just a minute by performing a simple experiment. Before we get started, remember, never touch or taste any chemicals. Handle glassware carefully, and always wear your lab coat and safety goggles. Also, make sure you have your science journal handy to take down notes and record results. All right, let's get going. Here's the equipment that I'm going to need. 4 test tubes and a test tube rack, a graduated cylinder, a wax pencil, some distilled water, egg albumin solution, 5% milk solution, 5% starch solution, and a dropper bottle of biuret solution. Biuret solution changes color in the presence of proteins. First, use the graduated cylinder to measure out 3 milliliters of each solution, which I've already done. Now, take the wax pencil and label the test tubes 1 to 4. Test tube 1 contains distilled water. Tube 2 contains the egg albumin solution. 3 contains the milk solution. And 4 contains the starch solution. Next, I'm going to add a dropper full of biuret solution to each test tube and mix it gently. Remember, the biuret solution will cause a color change if the substance is a protein. That's nice. The water and starch are blue, no color change. The egg albumin had a color change, and the milk solution had a slight color change. Now I'll take a moment to record the findings in my data table. Time to clean up and wash my hands. So the egg had a dramatic color change, but the milk solution only had a slight color change. Well, it was only 5% milk solution. What if I had a higher percentage, like 20 or even 40%? Wouldn't the color change be more noticeable? That's something I can test the next time. But right now, I need a protein boost. Milk or scrambled eggs? Decisions. Does anybody have a fork? Cellular respiration in plants. Everyone can tell when we've had a good workout. We're out of breath, we're tired, we're sweaty, and our muscles may be shaking. These are visible signs that the cells in our bodies are carrying out cellular respiration to give us the energy we need to ride, jump, swim, and play. Cellular respiration is a process that also occurs in other organisms, like plants. But how can we tell that cellular respiration is happening in a plant? We're going to answer this question in just a minute by performing a simple experiment. Before we get started, remember, never touch or taste any chemicals. Handle glassware carefully. And always wear your lab coat and safety goggles. Also, make sure you have your science journal handy to take down notes and record results. All right, here's what we'll need for this experiment. 2 test tubes labeled A and B with stoppers, a test tube rack, 20 milliliters of bromethymol blue, 5 germinating seeds, and 5 boiled seeds. When respiration occurs in a plant cell, glucose breaks down to form ATP. In case you're not familiar with the terms, glucose is a type of sugar, and ATP is a chemical that is used as energy within the cell. This process also releases carbon dioxide and water. Carbon dioxide is colorless, odorless, and really hard to detect in the lab. So we're going to use bromethymol blue as a carbon dioxide indicator. Bromethymol blue is a chemical that changes color when carbon dioxide is present. Be careful with this. It can cause stains on clothing, and if you get it into your eyes, immediately flush them with water. Now, I've already poured 10 milliliters of the bromethymol blue into each test tube. I'm going to take the 5 germinating seeds and place them in test tube A and put the stopper on it. The 5 boiled seeds go into test tube B. Put the stopper on this one too. I'll record the color of the test tubes in my data table. Then I'll come back in 24 hours to check the color. Okay, after 24 hours, it looks like the bromothiamyl blue is doing its job. There is definitely a color change in test tube A. I'll take a minute to write down my observations in the data table. All right, time to clean up and wash my hands. So here's the final data table. The germinating seeds in test tube A released carbon dioxide. We know this because there was a color change. But do we know why this is happening? Do you remember at the beginning of the lab we said that cellular respiration involved taking sugar and breaking it down? Seeds of plants are stuffed full of sugars. Breaking those sugars down releases carbon dioxide. This allows the plant to harvest energy necessary to produce roots, shoots, and leaves. Remember, there wasn't a change in test tube B. The boiled seeds did not release carbon dioxide, so no color change was observed. Using this same basic procedure, we could compare the rate of cellular respiration between different plant types by observing how long it takes for different test tube samples to change color. So plant cells are working out all the time. They really do lots of work that we can't even see. Whoa, slow down there, buddy. It's my camera time, not yours. Chlorophyll chromatography. Look at any well-kept garden and you'll see a wide variety of plants. Brightly colored flowers and healthy green leaves harness light from the sun. As you know, sunlight is a form of electromagnetic radiation. In the electromagnetic spectrum, visible light covers a range of different wavelengths. These wavelengths of light each produce a different color. Plants are made up of many different pigments, and each pigment reflects certain colors of light and absorbs other colors. That means that each plant pigment absorbs specific wavelengths of electromagnetic radiation. Is there a way to see the pigments that absorb sunlight in plant leaves? We're going to answer this question in just a minute by performing a simple experiment. Before we get started, remember, never touch or taste anything in the lab. Handle glassware carefully and always wear your lab coat and safety goggles. Also, make sure you have your science journal handy to take notes and record results. Okay, we'll get started by assembling the equipment that we'll need. A piece of coffee filter paper or chromatography paper, 15 milliliters of 95% ethyl alcohol, a pinch of sand, 2 toothpicks, a mortar and pestle, a 50 milliliter graduated cylinder, a 250 milliliter beaker, a test tube and rack, plastic wrap, a ruler, pencil, and scissors. I'm also going to need 5 to 10 fresh spinach leaves. First, I'm going to cut the spinach leaves into small pieces with the scissors. Now I'll put these pieces into the mortar and grind them up with a pestle for about a minute. I'll add a pinch of sand and 10 milliliters of the ethyl alcohol. We add the sand as an abrasive to help break up the spinach leaves into smaller pieces. The ethyl alcohol is added because chlorophyll dissolves in ethyl alcohol, but not in water. The ethyl alcohol is going to help us draw the pigments out of the leaves. Be careful with the ethyl alcohol since it's flammable. Keep it away from any open flames. And it's really important to be in a well-ventilated room too. Now I'll put this mixture into the beaker, cover it with the plastic wrap, and leave it for about 10 minutes. This will allow the ethyl alcohol to work on dissolving the chlorophyll. 10 minutes are up. Now I'll cut a thin strip of the paper, making sure it's longer than the test tube. We're going to create a color graph on the paper. I'll roll one end around the toothpick to hold it in place. This end goes into the test tube, and the toothpick rests across the top. The paper should not touch the bottom of the test tube. I'll take it out and measure 1 to 2 centimeters from this end of the paper and draw a line across it. With the other toothpick, I'm going to put a small drop of the liquid from the spinach mixture on the pencil line. By placing this drop on the line, we know where our pigment started and we'll be able to measure how far the pigments moved. Now I'm going to pour 5 milliliters of the ethyl alcohol into the test tube. The filter paper goes back into the test tube. I'll cover it with plastic wrap, leave it here, and wait 20 minutes. Our spinach solution contains plant pigments and ethyl alcohol. As the alcohol comes into contact with the paper, it will progress up the paper. This is due to the attraction between the molecules in the alcohol and in the paper. As the ethyl alcohol moves, it will take the dissolved chlorophyll pigments with it. We should start to see a pigment line on our paper. 20 minutes have passed, so now I'll remove the paper and let it dry. Now that the paper is dry, you can really see the different bands of pigment. Plant leaves usually have chlorophyll B and 2 or 3 other pigments. The more pigments they have, the more they're able to absorb light in every part of the light spectrum. I'm going to draw the different bands of pigment for spinach in my science journal. I'll label the lines with the name of the pigments. I used green spinach leaves, but what if we used yellow or red leaves? What colors would appear on the paper? Do plants that have more bands grow faster since they absorb more light? Maybe I'll try that next time. Oh, for me? You shouldn't have. These are definitely not spinach leaves. Comparing plant and animal cells. You have something in common with the towering trees in the forest and the green grass in your backyard. Cells. But are the cells of plants and animals alike? We're going to answer this question in just a minute by performing a simple experiment. Before we get started, remember, never touch or taste any chemicals. Handle glassware carefully. And always wear your lab coat and safety goggles. Here's what we'll need for this experiment. A compound light microscope, tweezers, a glass slide and cover slip, and a dropper bottle of iodine solution. I'm also going to need an onion and a prepared slide of human cheek cells. To use the compound light microscope, you need to start viewing the slide on the lowest setting and gradually move to greater magnifications. Use the coarse and fine adjustments to improve what you see through the lens. All right, let's get busy. First, I'm going to use the tweezers to pull a small piece from the onion. I want to try and make sure I get the thinnest piece I can, and I only need enough to fit onto the center of the glass slide. I'll add a drop of iodine to the onion. The iodine stains parts of the onion cells so that we can see features of the cell. I'll carefully hold the cover slip at a 45-degree angle over the iodine and gently lower it. There we go. Would you take a look at that? I can actually see the individual onion cells. That's cool. Looks like this magnification will work. I'm going to make a drawing of what I see in my science journal. Next, I'll take the prepared side of human cheek cells and look at it through the microscope. I'll make a drawing of what I see in my journal. My drawings show that we do have some things in common with plants. At least our cells do. We both have cells with a nucleus and cytoplasm, but plant cells also have a cell wall and central vacuole. So plant cells aren't completely like animal cells, and that's okay, because long hair just wouldn't look right on an onion. Or would it? I can see your roots. Earthworm dissection. When you think of recycling, you may not think of earthworms. But in a compost pile, earthworms can turn food scraps into great fertilizer. And earthworms' body systems work together to get the job done. What kinds of organs are found in an earthworm, and what do they look like? We're going to answer these questions by performing a simple experiment. Before we get started, remember, exercise caution when in the lab. Handle sharp items carefully, and always wear your lab coat and safety goggles. Also, make sure you have your science journal handy to take notes and record results. All right, here's what we're going to need for this experiment. Gloves, a dissection tray, dissecting pins, forceps, a scalpel, scissors, probe, and a hand lens. And of course, we can't get very far in an earthworm dissection without a preserved earthworm. First, I'm going to lay the earthworm in the dissection tray, ventral or flat side down. Now I'll use the hand lens to find the prostomium. That's the lobe that goes over the mouth. And this swollen band here is the clitellum. The clitellum produces an egg capsule which holds fertilized eggs until they mature. Now, I'm going to turn the earthworm over and make a shallow cut on the clitellum. I'll spread the incision using my forceps and pin down the sides of the earthworm with the dissecting pins. Now I'm able to see the aortic arches. These act as the earthworm's hearts. I'm going to check the other organs that make up the circulatory, digestive, excretory, and reproductive systems of the worm and make drawings and notes in my science journal. Time to clean up and wash my hands. I knew that earthworms and humans have different organs, but I guess I never thought about the body systems we share. We both have circulatory, reproductive, digestive, and excretory systems. Next time I can take a closer look inside the digestive system to see what the earthworm ate. The earthworm in our compost pile at school would have helped out with some old pizza crust, some apple cores, and a whole lot of Friday's special meatloaf. Extracting DNA. Deoxyribonucleic acid, also known as the three powerful letters DNA, is found in every living organism. Scientists remove DNA from the cells of plants and animals to study many things, from creating a hardier wheat crop to developing treatments for diseases. But how do they get the DNA out of the cells? We're going to answer this question in just a few minutes by performing a simple experiment. Before we get started, remember, never touch or taste any chemicals, handle glassware carefully, and always wear your lab coat and safety goggles. Also, make sure you have your science journal handy to take notes and record results. All right, let's get going. Here's what we're going to need. 2 cups of ice cold water, a pinch of salt, a pinch of meat tenderizer, 2 tablespoons of liquid dishwashing detergent, ice cold 95% ethyl alcohol, a 250 milliliter beaker, a test tube, a strainer, a stirring rod, a wooden stick, and a blender. We're also going to need a half a cup of a DNA source. In this case, we're going to use spinach. Why? Because I happen to like spinach. And the other choice was chicken liver. Ugh. I'm going to take the spinach and put it in the blender. Add a pinch of salt and the 2 cups of cold water. And I'm going to let it blend for about 20 seconds. The action of the blender and the salt will start to separate the cells in the spinach. Here we go. Now, using the strainer, I'm going to pour this lovely green substance into the beaker. It'll take a few minutes for the liquid to drip through the strainer. This liquid contains the cells. Now we're going to add the detergent to the liquid in the beaker. The dishwashing liquid is going to break down the cell and nuclear membranes. I'm going to stir it gently and let it sit for 10 minutes. Time's up! I'll take some of the liquid from this beaker and pour it into this test tube, filling it about a third of the way up. Remember, if any glass breaks, clean it up properly and carefully. Add a pinch of the meat tenderizer and stir gently. The enzymes in the meat tenderizer will break apart the proteins that wrap around the DNA. Next, I'm going to measure out about 2 milliliters of the cold ethyl alcohol. I'm going to tilt the test tube and slowly add the alcohol so that it forms a layer on top. You see these white stringy masses? That's the DNA. Spinach DNA in a few easy steps. I wonder if I use liver as a source instead, would I get more DNA? Maybe I'll try that next time. See you later. I'm going to clean up and wash my hands. Lichens up close. Many animal species rely on another species for their survival. This relationship also occurs in plants. Lichens live on solid surfaces, like rocks or trees. They contain a fungus and one or more types of algae. The fungus and algae rely on each other for survival. This is referred to as symbiosis. Although we can see differences between lichens with the naked eye, we can't necessarily see that they comprise two different organisms. So we'll set up an experiment that will help us. Before we get started, remember, handle glassware carefully and always wear your lab coat and safety goggles. Also, make sure you have your science journal handy to take down notes and record results. Let's get going. Here's our equipment and supply list. Samples of different lichens, scissors, forceps, a slide, a dissecting microscope, and some colored pencils. First, I'm going to cut a small piece of this lichen and place it on the slide. Okay, slip this under our microscope, and let's see what we've got. I definitely see different colors. I'll take a minute to draw what I see in my science journal. Okay. Now at this point, I'm going to follow the same steps for the other samples and make comparisons when I'm done. The lichens I studied were all different in color and shape, and they all had different amounts of green material. That's because there were different combinations of algae and fungi in each one. I wonder if similar plants will look the same as lichens. Would moss look the same through a microscope? Maybe we'll try that next time. Who knew such a small organism could be so complex? Natural selection in bacteria. Washing your hands is one of the best ways to avoid spreading diseases. Antibiotics and soaps and medicines kill bacteria that can cause illnesses. But they can also kill beneficial bacteria at the same time. Not all bacteria are harmful. But can bacteria become resistant to antibiotics? And do we really want to eliminate all bacteria from our lives? We're going to answer these questions in just a few minutes by performing a simple experiment. Before we get started, remember, handle glassware carefully. Be careful when working with an open flame, and always wear your lab coat and safety goggles. Also, make sure you have your science journal handy to take down notes and record results. All right, let's get started. Okay, here's what we'll need for this experiment. Gloves, a marker, 2 nutrient agar petri dishes, Bunsen burner, inoculation loop, and forceps. We also need a bacteria culture and an antibiotic disc. My hypothesis is that an antibiotic disc will kill all the bacteria in the surrounding area. Let's begin. I'm going to label the bottom of one Petri dish A and the other Petri dish B. Dish A will be the one with the antibiotic disc, and dish B will be my control. Now I'm going to sterilize the inoculation loop by using the Bunsen burner. Be careful around an open flame. I'll use the inoculation loop to get a small amount of bacteria culture. This is applied to the Petri dishes in a zigzag pattern, but you don't want to break the surface of the nutrient agar. Next, I'll use the forceps to pick up an antibiotic disc. The disc will go into Petri dish A. I've covered both Petri dishes and set them aside. I'm going to check them at the same time 3 days from now and record my findings in my science journal. Time to clean up and wash my hands. Well, 3 days have passed, and you can really see a difference in the Petri dishes. In Petri dish B, the bacteria covered the dish, just as we expected. There was no antibiotic present to inhibit growth. In Petri dish A, the antibiotic disc killed most of the surrounding bacteria, but a few bacterial colonies grew even in the presence of the antibiotic. The fact that some bacteria grew, even with the antibiotic disc, means that there are some antibiotic-resistant bacteria out there. What would happen if I used another bacteria culture? Would the individual variances in the bacteria lead some to be more resistant? And are they ones that reproduce? Are they more fit for that situation? Over time, would the resistant colonies continue to grow and thrive? It's clear that bacteria adapt and become resistant to antibiotics. Those single-celled organisms sure are tough. And all species adapt to their environments. Otherwise, species would go extinct. You know what they say. Adapt, migrate, or die. Onion root tip. If you take a close look, a very close look at this person or this dog or this tree, you'll see billions, maybe trillions of cells at work, growing and repairing damage. Each new cell is a building block of life, but where do the new cells come from? We're going to answer this question with a simple experiment. We only need a couple of items, a microscope and this prepared slide of an onion root. Be careful with the slide. If it breaks, clean up and throw away the glass properly. Oh, and I'll definitely need my science journal for notes and observations. Let's get started. A few quick adjustments on the microscope. Okay. Here's the onion root in various stages of cell development. I'm going to draw the different phases and label as much of the cell parts that I can in my science journal. Okay, I think I found all the phases of mitosis. Interphase, prophase, metaphase, anaphase, telophase, and here's one picture of a cell in cytokinesis. That's where the nucleus of a cell finishes dividing and the rest of a cell grows larger and divides in two. This cycle is continuous, and that is why we can see different phases in different areas of the onion root tip. I could tell the cell was in interphase because I found the entire nucleus inside. In this picture, the chromosomes lined up at the center, so I knew the cell was going through metaphase. And I finally figured out why we use an onion root. Because the root is constantly growing. New cells come from pre-existing cells. The cells in the tip of the root are going through mitosis all the time. That is, until this onion grows up big enough to get chopped into some salsa. Yeah. That one had a little zip to it. Osmosis in cells. Water is important for our survival. At the most basic level, cells use water to transport the chemicals needed to sustain life. So how does water move through cells? We'll be able to answer this question in just a minute after we perform a simple experiment. Before we get started, remember, never touch or taste any chemicals. Handle glassware carefully, and always wear your lab coat and safety goggles. Okay, besides a science journal, here's what we'll need. A microscope, slides and cover slips, forceps, an eyedropper, an elodea leaf, some distilled water, and 10% sodium chloride solution, that is, salt water. Elodea is a freshwater plant. We're going to examine the elodea leaf to see how water moves in and out of the cells. Water enters and leaves cells by osmosis. Osmosis is the movement of water from areas of high concentration to areas of low concentration. When the movement of water is balanced, you can see the elodea cells are bright green and the cytoplasm fills the cells. If the cells are surrounded by a hypertonic solution, a solution with a higher solute concentration than normal cells, water will leave the cells and the cells will shrink and collapse. Let's see if this happens in our experiment. Use the forceps to get a small elodea leaf. Put a drop of distilled water on the leaf and place the cover slip on top. Be careful with the glass slide. If it breaks, clean it up properly. Take the wet mount and place it on the microscope stage. Focus and adjust the microscope. What do you see? Is the water entering or leaving the cells? I'm going to take a second to draw what I see in my science journal. We're using distilled water, so the water is rushing into the cells. But what if we use the salt water? What do you think would happen? Let's try. Again, use the forceps to get a small elodea leaf and place it on the microscope slide. Put a drop of salt water on the leaf and carefully place the cover slip on top. Take a look. See what's happening? The water is leaving the cells and moving towards the area of lower water concentration, the salt water. This is causing the cell's cytoplasm to contract and pull away from the cell wall. Pretty neat, huh? Remember to clean up and wash your hands. So water is vital for cells to survive. You don't want droopy cell walls, so remember to water your plants. And don't forget, your cells need water too. Planarian behavior. Animals exhibit a variety of behaviors, such as staring down their enemies or using sounds to attract a mate. You can learn a lot just by watching an animal. But can animals learn new behaviors? We're going to answer this in just a minute by performing a simple experiment. Before we get started, remember, never touch or taste anything in the lab. Handle glassware carefully, and always wear your lab coat and safety goggles. And make sure you have your science journal handy to take notes and record results. First off, our materials list: a stopwatch, a syringe without the needle, a petri dish, and a small paintbrush. We're also going to need a planarian and some pond or spring water. Planarians are flatworms that live in ponds and streams. We're going to study the planarian because it shows innate behaviors it was born with. But it also is capable of learning new behaviors. So let's see if this animal can learn new behaviors. I'll take the paintbrush and put one planarian in the petri dish with the water. I'm going to let the Planarian adapt to the dish for about 5 minutes. Okay, so now we can get started. Using the syringe, I'm going to blow a puff of air on the Planarian every 60 seconds for 10 minutes and observe how it reacts. I've set up this data table to record my findings. I've got the trials listed here, and I'm going to use the following letter codes for my observations. C is for contracts, T is for turns away, and N is for no response. Ready? Here we go. All right, we've been at this for 10 minutes, and I've got some good data. Now it's time to clean up and wash my hands. From the data, we can see that the Planarian contracted and turned away in the beginning, but towards the end, there wasn't a response. I wonder if it got used to the puff of air, knew that it wasn't in danger, so it didn't react. Did it learn a new behavior? Maybe there's more to these little guys than meets the eye. Whoever said you were simple-minded, huh? Testing for lipids. Time for lunch. I've got my carbohydrates. The chicken on my salad is the protein. But do I have a lipid? And how will I know if something is a lipid? We'll answer this by performing a simple experiment. Before we get started, remember, never touch or taste any chemicals. Handle glassware carefully. And always wear your lab coat and safety goggles. Here's the equipment and materials we'll need for this lab. 3 test tubes and stoppers and a test tube rack. A wax pencil, water, vegetable oil, and Sudan 3 dye solution. We're going to use the Sudan 3 dye to identify which liquid is a lipid. Say that 3 times fast. The Sudan 3 solution will mix with a liquid lipid. We'll also need to make a data table like this one to record our findings. Now that we've got everything, let's get started. First, I'm going to use the wax pencil to label the 3 test tubes from 1 to 3. As you can see, I've already added 2 milliliters of water to test tube 1, 2 milliliters of vegetable oil to test tube 2, and 1 milliliter of water and 1 milliliter of vegetable oil to test tube 3. Now I'll add 3 drops of Sudan 3 to each test tube. I'll shake each test tube well and allow them to settle. Sudan 3 is a large organic molecule that likes fats. What do you think will happen in the different test tubes? The Sudan 3 sat on top of the water in test tube 1. It didn't mix with it. The Sudan 3 mixed with the vegetable oil in test tube 2, so we know that's a lipid. In test tube 3, the Sudan 3 dye mixed just with the vegetable oil that separated from the water as the liquid settled. I'll record the findings in my data table. Because Sudan 3 is an indicator that identifies the presence of lipids or fats in liquids, this same procedure can be used to test other food types. I'm getting hungry. Time to clean up and wash my hands so I can get back to my lunch. So you see, lipids are a natural and essential part of many of the foods we eat. Oils and fats are both lipids. Maybe for my lunch, I'll stick with some butter for my roll. Testing for starches in food. We need a balanced diet to give us the energy we need to work and play. Starches serve as a quick source of energy. But how can you tell if food is a starch? We're going to answer this question in just a few minutes by performing a simple experiment. Before we get started, remember, never touch or taste anything in the lab and always wear your lab coat and safety goggles. Okay, let's get started. All right, for this experiment, we're going to need a small bottle of iodine and a paper towel. We're also going to need 10 small samples of different food. I'm going to place all the samples of the food on the paper towel. Then I'm going to take a drop of iodine and place it on each sample of the food and see if it changes colors. Iodine is a reddish-brown liquid that turns blue or black on a starch. Be careful with the iodine. It can stain your clothing. After all my observations, I'll record the findings in my data table. All right, here we go. Okay, all done. I'll clean up and wash my hands. So just as I suspected, the potato, the pasta, and the corn all turn black because they're starches. But I also had some surprising results. Looks like I need to make my meals a little more balanced. Examining the body system of a grasshopper. With long legs and a body made for hopping, grasshoppers sure can move. You've probably heard their distinct sounds or seen them in your garden. They may be small, but there's a lot going on inside their little bodies. Ever wonder what organs are found inside a grasshopper? Only one way to find out. That's right, by performing a simple experiment. Before we get started, remember, never touch or taste anything, and always wear your lab coat and safety goggles. Also, remember to have your science journal handy to take down notes and record results. Here's what we're going to need. Gloves, dissecting tray, scissors, probe, and forceps, an eyedropper, and a small beaker of water. I'm also going to need a preserved grasshopper. Thanks for donating yourself to science, my little friend. All right, let's get going. The first thing I'm going to do is make a sketch of the grasshopper in my journal, and then I'll label all the external parts I see. As I look at the abdomen and search under the wing, I find the tympanum. It's basically the grasshopper's eardrum. Oh, and here are the spiracles on the side of the abdomen. They're the tubes that allow the air in so he can breathe. Now I'm going to take the scissors and carefully remove the wings and legs. Then I'm going to cut each side gently from the head towards the abdomen and remove the exoskeleton. So our skeletons are on the inside and theirs are on the outside. Hence the name exoskeleton. Makes sense to me. Now that I've removed the exoskeleton, I can really get a closer look at the internal organs. To keep the organs from drying out as I'm working, I'll use the eyedropper to put a little bit of water on them. Now I'll look for some of the organs and sketch them in my journal. Time to clean up and wash my hands. I could identify most of the organs in the grasshopper, but some were really too small to be seen without a microscope. Maybe I'll try that next time. It's incredible that such tiny parts make up the systems that help the grasshopper live, breathe, and jump.